Okay, so the topic of this video are some of the basic characteristics of bacteria. So let's get started. Well, you might know that there's two big broad categories of cells, the prokaryotes and the eukaryotes. You know, when you look at this picture right here, you can see, wow, the eukaryotes are just much larger. Well, what are eukaryotes? Well, these are cells that belong to protista and fungus and plants and animals. But our video today is focusing on prokaryotic cells, the bacteria, and then the group of called archaea. Now we're mainly focusing on the bacteria today, but bacteria and archaea are examples of prokaryotes. So let's talk about them now. So when we focus on the cell structure of bacteria, probably their most defining feature is that they're prokaryotes. They lack a nucleus and other membrane-bound organelles, organelles that you might be familiar with if you studied cell parts. They lack, for instance, a nucleus, they lack mitochondria, they lack the rough and smooth ERs, they lack Golgi bodies. But what do they have then? Well, they're cells, so they're going to have genetic material. They're going to have a chromosome in what we call the nucleoid region. Now it's all wound up, you see in this picture right here. It's not in the nucleus, it's just kind of floating around in like the middle of the cell in the area kind of called the nucleoid region. They also have these other segments of DNA called plasmids, and these are ring-shaped structures separate from the main chromosome that has genes on them that they can actually transfer to one another. We'll talk about that in a little more detail in a moment. Well, another organelle they have are ribosomes. Ribosomes, you might know, are organelles that help to create proteins. And here's a ribosome, and as it's creating a protein, it's actually gathering amino acids and linking them into a chain. Here we have a bunch of amino acids being gathered, and once that's finished, the amino acids will ball up and twist and fold and wrap into a protein. Well, that's a job performed by the ribosome to create proteins. Well, we come back to our diagram here. Notice they have a long whip-like tail on them called a flagellum. The plural version would be flagella. So sometimes a bacteria will have a single flagellum. Sometimes they'll have multiple flagella. But when they whip their flagellum back and forth, it propels them through their watery environment. It's how they move around. Notice going around the outside of the cell are these short hair-like extensions called pili, the singular version being pilus. And these things will actually help to attach and anchor the bacterium onto whatever it is they're growing upon. They also have another function which is kind of interesting. Now, here's their other function. Here's a bacteria cell, and the process called conjugation, where DNA, segments of DNA, can actually be swapped and exchanged from bacteria cell to bacteria cell. Well, here's a bacteria, and in black is the main chromosome floating around in the nucleoid region. In red, there's the plasmid, and sticking out, there's what's called a sex pilus. Now watch what happens. Another bacteria cell will actually combine and join with the bacteria on the left. Watch the red plasmid. The plasmid is duplicated, and that plasmid will actually be transferred through the sex pilus into the neighbor on the right. And so when they disconnect, you can see that conjugation is a way for bacteria to obtain advantageous genes and segments of DNA from their neighbors that they can then use throughout the rest of their lives. Okay, so we'll come back to our diagram here. Now let's focus on their three outer layers to them, starting with the innermost, the layer in green, the plasma membrane. This is the phospholipid bilayer. You might know that it is selectively permeable. It allows molecules to pass in and out of the bacteria cell. Well, the next layer in gold is one of the defining features of a bacteria, their cell wall, the stiff layer that helps them maintain their shape, made from a combination of amino acids and sugars that we call peptidoglycan. I say one of their defining features because the cell wall is unique. This type of cell wall is unique to bacteria. You might know plants have a cell wall, but it's different from the bacteria's. You might know fungus have a cell wall, but it's different from bacteria. So the cell wall made of peptidoglycan is a pretty unique structure in bacteria. Well, notice there's another outer layer called the capsule, the slimy outer coating that actually helps the bacteria stick to surfaces and prevents it from drying out. So we actually see three outer layers to them: the plasma membrane, the cell wall, and the capsule. And it's worth noting that even some bacteria can produce what's called an endospore, a cocoon, to actually protect their DNA during times of stress and harsh environments. So in this situation here, here's a bacteria cell being exposed to a high amount of UV radiation. Well, in this case, an endospore might begin to grow inside of the cell and actually cover and protect the nucleoid region in the DNA of the bacterium. Now, over time, this bacteria cell could actually be destroyed, but the endospore protects the DNA inside, and these things can last decades upon decades. And let's say that over time, the environmental conditions change and the area is not exposed to as much UV radiation anymore. Well, the endospore could kind of come out of this dormant stage and actually regrow into the bacteria cell so it can continue on its existence. It's a neat little adaptation that some bacteria have in order to protect themselves in times of environmental stress. Okay, let's kind of change directions here and talk about the various shapes of the bacteria cells. Three basic shapes. You have the rod-shaped bacteria. Bacillus is the singular version and bacilli is the plural version. And like the picture shows, these are rod-shaped cells. Then there are the spherically shaped cells. Coccus is singular, cocci is plural. And then there are the spiral-shaped bacteria cells, the spirillum. Spirillum is singular and spirelli is plural. And so when we combine these shapes with the next part, so the next part being that many bacteria grow in colonies, and when you see prefixes in the name of a bacterium, it actually gives you a lot of information. For example, the prefix diplo means that the bacteria are found in pairs, and this bacteria right here is called a Diplococcus bacteria. Well, I hope you see why the word coccus is in its title. You can see the spherical shape. Diplo because there are two cells. If you see the prefix strepto in front of the name of a bacteria, these are bacteria that are found in chains. And so this example right here, Streptobacillus bacteria, well, you can see from the picture the bacillus is in reference to the cells are rod-shaped, strepto because there are chains of rod-shaped bacteria cells. And then there's the prefix staphylo, which means grape-like clusters. So when you see, for instance, Staphylococcus bacteria, well, you can see why they're called coccus because they're round and spherically shaped, and staphylo because they kind of look like cluster of grapes. So when you see these prefixes, it gives meaning to the bacteria, the type of bacteria. Let's talk about how bacteria reproduce. Well, they reproduce asexually through a process called binary fission, where one cell will split into two cells. In my animation, here's a cell, and you can see the round genetic material in the nucleoid region. Notice how the a copy of the DNA is created. As the cell goes through binary fission, it'll start to enlarge, and the pieces of DNA are pulled to opposite ends. And then right down the middle, the cell begins to divide until they are separated into two cells. And so each of these cells are identical in their DNA because in asexual reproduction, you make clones, exact duplicate copies. Now, this is a bit of a problem for them because organisms that reproduce asexually tend to suffer from less genetic diversity. After all, they're exact duplicates. So if something would harm the cell on the left, chances are it would harm the cell on the right. But there's a neat little solution to this. The solution is what we talked about briefly earlier called conjugation. In conjugation, here's the main chromosome in the nucleoid region of our bacteria cell, and in red, here's the plasmid. And poking out to the right, there's the sex pilus that we mentioned earlier. So the cell on the left is now going to connect to the cell on the right. Now watch the plasmid in red. The plasmid is duplicated, and one of those copies is transferred through the sex pilus into the neighbor on the right. And so when they disconnect, when they separate, you can see that the bacteria now have exchanged segments of DNA. So is conjugation, first of all, is it a form of reproduction? And the answer is no, but it's a solution to their problem, the problem of having less genetic diversity. Conjugation solves that because they're able to obtain genetic diversity from their neighbors by swapping and exchanging segments of their plasmids in their DNA like this. Okay, well, let's change direction here and let's talk about how do they use oxygen and how do they make their energy? How do they perform respiration? Well, here's 3 test tubes and each of them is filled with like a nutrient broth. Focus on the test tube on the left. Let's put a cap on it. Now, just beneath the cap, you figure there's a little more oxygen. At the bottom of the test tube, there's probably very little oxygen. Well, over time, what if a bunch of bacteria begin to grow just underneath the cap where there's more oxygen? Well, these bacteria would probably be called obligate aerobic bacteria. They are obligated to live in oxygen, and that's what the word aerobic means, with oxygen. Well, how do they use the oxygen? They use the oxygen to make ATP, the energy molecule. They actually perform cellular respiration. Now, when you hear that, you might think, well, wait a minute, how do they do that? In order to do cellular respiration, you need mitochondria. Bacteria don't have mitochondria, and that's correct. But they are still able to perform cellular respiration because they have the necessary enzymes to do cellular respiration in their cytoplasm and in their cell membrane. So they're able to make ATP with oxygen, and that's why they're aerobic bacteria. A good example of this bacteria are the bacteria that can cause tuberculosis. These are bacteria that will infect the person's lungs, and of course, living in your lungs, they're exposed to lots of oxygen. Well, let's look at tuberculosis in a little more detail. Now, if we look at this chest x-ray of a person's healthy lungs, here are the lungs right here blinking. Notice how the lungs appear just black, because after all, they're filled with air and it doesn't really show up on an x-ray. And that's what a healthy chest and healthy lungs would look like in an x-ray. But in this picture, here's a chest x-ray of a tuberculosis patient. See this cloudy area right here? How come it's cloudy? Well, it's cloudy because it's filled with blood and pus, and they're constantly coughing this up, and it's a sign that the person's quite ill because of the bacteria infection that they have living inside of their lungs, the bacteria that causes the disease tuberculosis. Okay, let's come back to our 3 test tubes and focus on the middle test tube. Again, let's add a cap. So you figure just beneath the cap there's more oxygen versus at the bottom of the test tube. Now, what if bacteria begin to grow, but they all tend to be concentrated near the bottom of the test tube where there's less oxygen? Well, these bacteria would probably be called obligate anaerobic bacteria. They are obligated to not live in oxygen. They can't survive the presence of oxygen. So what actually happens is, in order to make their energy, they perform fermentation. They perform the anaerobic cellular process called fermentation, and that's how they make their energy in order to survive. Well, you know, here's something maybe you've noticed at a grocery store. Have you ever picked up a can of food and noticed that the can was swollen or puffy? I hope not. But if you have, that's probably a sign of some anaerobic bacteria growing inside of the can. There's no oxygen in the can, so if any bacteria are going to grow in there, they have to be anaerobic. And because of their actions, they give off gases and it causes the can to swell and become puffy. And so a swollen, puffy can might be a sign that there's anaerobic bacteria growing inside. If you've ever heard of the type of food poisoning called botulism, It's a type of food poisoning caused by anaerobic bacteria. So if you ever see swollen, puffy cans, don't buy them. Report them to customer service at the grocery store and then move on. But don't buy them and eat them. Okay, let's come back to our 3 test tubes one last time. The test tube on the right, let's add a lid. So you figure just beneath the lid, there's more oxygen versus at the bottom of the test tube. And over time, what if bacteria begin to grow, and they grow all throughout the test tube? Although there's more bacteria accumulating near the top where there's oxygen, there are still some growing near the bottom where there's little, if any, oxygen. Well, these are the bacteria that are called the facultative anaerobic bacteria, which means they can live with or without oxygen. If there's oxygen present, they're able to do cellular respiration. But if there's not oxygen present, then they perform fermentation in order to produce the energy that they need to survive. So bacteria, there's a wide variety of ways in which bacteria can obtain their energy and survive. Okay, so there you have it. If you want to challenge yourself, pause the video, try to answer these questions, and I hope you found this video helpful. Thanks for watching.